Hello, and welcome to the Chapter 14, Part 2 lecture on the exceptions to Mendelian rules. You should use the information in this lecture to complete the Chapter 14, Part 2 guided notes, which of course you should complete before you come to class. In Part 1 of this chapter, we discussed the contributions of Gregor Mendel, the father of genetics. He worked with pea plants, and by breeding pea plants together very carefully over many generations, and by keeping very thorough notes, Mendel was able to figure out the basic patterns of inheritance used by living things. He focused on the relationship between dominant and recessive alleles. And it just so happens that all of the characteristics that he studied over the years just so happened to play by those dominant and recessive patterns that we discussed in the first part of the chapter. Now, not every characteristic in pea plants plays by these rules, but he didn't notice that because he didn't happen to choose those characteristics. So Mendel discovered a number of alleles that play by the dominant and recessive pattern, and he very thoroughly described that pattern. In reality, though, not all alleles play by this specific pattern. There are many different types of relationships that alleles can have and different kinds of interactions. In fact, it's actually really difficult to find characteristics in the human system that do play strictly by these rules. There are a lot of traits that we used to attribute to this. For instance, you might have heard that tongue rolling is controlled by a single gene, and you're dominant if you can roll your tongue, and you're recessive if you're not. Turns out it's not that simple. So let's learn about some exceptions to Mendelian rules. Some alleles are neither dominant nor recessive to one another, but instead are expressed equally when they are inherited together. We refer to this as incomplete dominance. A great example of incomplete dominance exists in snapdragons. You may have seen these flowers growing around campus in the early spring. There are red snapdragons and white snapdragons. If a snapdragon is red, that means it has inherited two red alleles from both parents. It's homozygous for that trait. If the snapdragon is white, it has inherited two white alleles from both parents. It's homozygous for that trait. But if you breed red and white snapdragons together, you get neither red nor white offspring. In fact, instead, you're going to get pink offspring. The pink offspring have inherited one red allele from one parent and a white allele from the other parent. They are heterozygous for that trait. Because these alleles are incompletely dominant to one another, the heterozygote is an intermediate between the dominant and recessive forms. So you get a blending of those two forms and you end up with pink flowers. If you extend this and take two of those pink flowers and breed them together, you'll get 1/4 of the flowers being red, 1/4 of the flowers being white, and half of the flowers being pink in the next generation. An example of incomplete dominance in the human system is achondroplasia. Achondroplasia is the most common form of dwarfism. This is caused by an allele that is considered to be dominant in many textbooks, although I think you'll see that it really plays more by the incomplete dominant pattern. This allele affects the growth of the embryonic skeleton, so individuals who inherit this allele tend to be short in stature. This especially affects the long bones, so the length of the arms and the legs. This gene plays by the incompletely dominant pattern because heterozygotes who inherit a gene for dwarfism from one parent and a gene for a normal skeleton from the other parent do tend to live for a number of years. They will be short in stature, but they can live a relatively normal life and can even have children and pass this gene on. On the other hand, individuals who inherit two of the dwarfism genes from both parents don't tend to live very long because having two copies of this allele just affects that skeleton to such a degree that the body can't form properly. So really, this gene acts like an incompletely dominant gene. Another interesting example of incomplete dominance in the human system is sickle cell anemia. Sickle cell anemia is a blood disorder that affects the shape of the hemoglobin protein inside the red blood cell. So in here you can see those hemoglobin proteins. The hemoglobin proteins carry oxygen for the red blood cell, and it helps give that red blood cell its shape. Here you can see the normal shape of a red blood cell. It's called biconcave. They are little discs with a dimple on either side, and this shape makes them very, very flexible and allows them to go around all the corners in some of these very tiny blood vessels. Individuals with sickle cell anemia have a point mutation in the gene for making hemoglobin. So one little letter in that DNA sequence has been changed, and that affects the shape of the protein so that the cells containing that mutated hemoglobin protein will sickle and take on this really strange kind of crescent moon shape. Now, those crescent moon shaped cells aren't as flexible as the normal ones. They tend to kind of clog up in the junctions of the blood vessels, and this can cause swelling and pain. They also aren't as good at carrying oxygen as the normally shaped cells. Individuals who are homozygous for the normal hemoglobin allele will have normally shaped red blood cells. Individuals who are heterozygous for this trait, who have one normal hemoglobin gene and one sickled hemoglobin allele, will have cells that are affected slightly by that allele. And so they will carry some abnormal hemoglobin, although it won't change the shape enough to really affect function. So heterozygotes pretty much have normal red blood cells. Individuals that inherit two sickled alleles that are homozygous for this trait will have the sickled cells. Sickle cell anemia is an interesting condition because although it does cause a blood disorder in those who inherit it, it also confers a benefit. Sickle cell anemia is found in the largest proportions in peoples that live near the equator. And near the equator is where we find the highest incident rate of malaria. That's where the mosquito that carries malaria tends to live. Here we can see some red blood cells that have been infected by the malaria parasite. This little protozoan gets inside those red blood cells and feeds on them, and this can cause horrible fevers, horrible flu-like symptoms, and malaria can even be fatal. So what does this have to do with sickle cell anemia? Well, if an individual is homozygous for normal red blood cells, they can catch malaria. If, however, an individual is heterozygous for sickle cell anemia or has sickle cell anemia, they cannot catch malaria. So although the sickle cell anemia can cause the anemia, can cause the sickling of the cells, it also changes the red blood cells just enough so that that little parasite can't get in there. So it gives them a resistance to malaria. Because it confers this benefit to the individuals, it tends to stay within family lines. Individuals who are heterozygous for this have a tendency to have more children, or they at least have the opportunity to have more children, and pass this gene on. Another type of relationship that can exist between two alleles is codominance. Codominance also involves the blending of the alleles in the heterozygote, but in a slightly different pattern of expression than we saw in incomplete dominance. Let's look at camellias as an example. Camellias are flowers that look a little bit like roses. There are white camellias and there are pink camellias. If you breed white and pink camellias together, instead of getting a complete blending of the alleles, a light pink flower or something like that, you actually get a white and pink spotted flower. So what's going on there is that in some of the petals, the white allele is dominant, and in other petals, the pink allele is dominant. So one kind of takes over in one area, and in other areas of the flower, the other one takes over. So we say that these alleles are codominant. They're both dominant, just in different parts of that flower. Another factor that can affect genetic outcomes are the presence of multiple alleles. So far, we've really just been talking about traits that have two alleles, a dominant and a recessive, or two that are incompletely dominant or codominant to one another. But many different genes have more than two alleles for that particular trait. As an example, let's look at eye color in fruit flies. Now, I don't know how much time you've spent looking at the eyes of fruit flies, but they're really quite striking, and they can occur in a variety of different colors. Red is the most common color, but they can also be purpley, pinky, yellowy, brownish, even white in color. And this occurs because there are many alleles for fruit fly eye color. And those alleles all have different kinds of relationships with one another. Some are dominant to others, some are recessive to others, some are codominant, incompletely dominant, and so on. So this makes making predictions about fruit fly eye color a little bit more complicated. In part one of the chapter, I had you examine your ear shape, and I had you try to determine whether you had free earlobes or attached earlobes. Well, although those phenotypes are the most common for earlobe shape, there are many different phenotypes that can exist for the shape of your ears. There are lots of different intermediate shapes. This occurs because ear shape isn't actually governed by a single gene with two alleles. It's actually what we call a polygenic trait. The shape of your ears is governed by several different genes, and those genes can have different alleles. So when you count up all the interactions you can have from those genes and alleles, you actually get a wide range of phenotypes that can occur. Another really great example of a polygenic trait is human skin color. Now, when we talked about albinism, we talked about melanin a little bit. Melanin is this dark pigment that's laid down in the skin, and in general, the more melanin you have, the darker your skin tends to be. But there's more than one type of melanin, and it turns out that there's actually three different genes that govern how much melanin is made, which type is made, and how it's laid down in the skin. So this is a polygenic trait that's governed by three separate genes. To complicate things even more, each of these genes has two alleles, a dark allele and a light allele. So depending on the combination of the alleles that you get for these genes, that's going to determine the skin color that you have. Here we have an interesting Punnett square that shows some of the combinations you can have. For instance, individuals who inherit all six dark alleles for all of those genes will have the darkest type of skin. Individuals on the other end of the spectrum that inherit the light allele for all of those genes will have the lightest, palest type of skin because they have the least amount of melanin. But in between, you have this wide range of skin colors and types that can exist, and that's very typical of a polygenic trait. Some genes only code for a single enzyme found inside a single type of tissue. So mutations in a gene such as that are only going to have a limited effect on the body. Other genes, though, are pleiotropic in nature, and that means that they can affect multiple traits within a body and thus have multiple phenotypic effects. We talked about this a little bit with master genes back in chapter 18. Master genes, because they are in charge of other genes, will affect multiple traits at a time. But genes can be pleiotropic if they affect a trait that's found all over the body. So for instance, let's say a gene affects how connective tissue forms. Because connective tissue is found throughout the body, a mutation in a gene that affects connective tissue can cause problems all over the body. We say it has multiple phenotypic effects. Another interesting example of pleiotropy occurs in certain breeds of cats. It's not uncommon for cats with really light or pure white fur and light blue eyes to also be deaf. And sometimes you'll have a cat that has heterochromia. That means they have two different colored eyes. And on the side of the head with the blue eye, they will also be deaf in that ear. So what's the connection between fur and eye color and deafness in this breed? Well, it turns out that the gene that controls the pigment in eyes and in fur also affects the formation of the inner ear. This is a gene that has a pleiotropic effect. It affects more than one characteristic. So the white fur and the blue eyes indicates that that gene is not active, that pigment is not being laid down in the fur or in the eyes. That's why they are blue in color. And because that gene is not active, it also means that it's not forming the inner ears properly. The environment in which an individual lives can also affect gene expression. We talked about this already a little bit back in chapter 18 when we discussed how diet can affect the epigenome that turns genes on and off. Let me give you another example of environmental influence. Here we have two interesting little breeds of animal that have similar fur patterns, a Himalayan rabbit and the Siamese cat. In both of these animals, you'll find dark fur around the muzzle and the ears and the legs, the parts of the body that stick out from the core of the body. And you'll find lighter fur on the core of the body. Well, why is that? Well, it turns out that these animals have a gene in the skin and in the fur that lays down pigment in the fur. But that gene is only active when it is colder, a little bit cooler than body temperature. So that gene is only active in the parts of the body that stick out from the body a little bit. So the ears are a little bit cooler than the core of the body, so there's more pigment being laid down there. All right, guys, you did it. You're finished. Make sure you complete your notes thoroughly, complete the year turns, and bring them to class with you, because we're going to do some genetics problems with this information. See you soon.